Introduction of luciferase and luciferin genes via Agrobacterium tumefaciens to Kentucky bluegrass. GMOs have become a relatively common part of our society. The most common GMOs are food. Vegetables and fruits are normally genetically modified to grow larger, have more nutritional value, and to be resistant to insects. Using GMOs to address social issues has been a newer approach over the years. During this project, we decided to address the issue of poorly lit walkways. This problem could be easily resolved by using genetically modified grass that glows at night. By lining sidewalks and other walkways that are normally not lit well at night, this would help reduce anxiety of walking alone at night and potential injuries. We chose this particular project in order to help make Morgantown safer, because no one likes to walk home alone at night. In our project, 2 genes will be introduced to Kentucky bluegrass to make it glow in the dark. These 2 genes, luciferase and luciferin, code for an enzyme and a substrate that can be found in types of bacteria and plankton, such as dinoflagellates, that make organisms glow. This particular issue has never been attempted to be resolved using GMO grass species before. However, recently a certain type of fish has been genetically modified to glow. These zebrafish, called glowfish, have been genetically modified by transforming the fish with several GFPs, or green fluorescent proteins, where they were still an embryo. As technical as this may sound, these fish are available to be commercially sold and kept as pets and have become quite popular. Unlike the glowfish, however, our grass will be useful to the community as a whole instead of just children afraid of the dark.
The gene we modified comes from phytoplankton in the ocean called dinoflagellates that has bioluminescence in a bluish-green color. The color of the bioluminescence can change. It simply can match the color of the excited luciferase in the reaction. The luciferase can affect the color by an amino acid being switched in the luciferase and lead to differences in the emission spectrum. Bioluminescence may not be a constant thing. It could happen in brief flashes due to changes in a cell's organelles or differences in pH levels. Light from these reactions come from the reaction at the excited state, when a photon of green light is released. The excited state does not last for any amount of time, so all of this is done very quickly. The actual emission of bioluminescence is quick and occurs in many steps. Bioluminescence is a variation of chemoluminescence, or a chemical reaction where light is produced. Luciferin and luciferase are 2 required chemicals of bioluminescence. Luciferin is a substrate where the light is actually produced. Luciferase is the enzyme that, when reacted with the luciferin substrate, forms light.
The specific promoter we used is chalcone synthase. It showed in the pigmented parts in most grass species. It is the UV light responsive, which is good because the genes will begin to form during the day, so they will be visible during the night. This modification will make walking at night safer, especially for those who do not see as well. The lighted pathways will guide people in the dark when walking along the sidewalks, as well as keep people alert and on track to their desired location. It does not hurt that glowing grass would look pretty cool.
Our organism is Kentucky bluegrass because it is a common type of grass and easy for homeowners to grow. It is also perennial, and it grows in all 50 states. To insert the specific genes we wanted, we chose to use Agrobacterium tumefaciens, a plant pathogen. Many GMOs that have been released commercially have used the pathogen and work successfully and safely. We chose this technique over microparticle bombardment because it is much cheaper and can achieve single copy insertions. EcoRI is one of the most commonly studied restrictive enzymes and breaks into the GAATTC sequence. This is the place where we want to insert our desired genes for luciferase and luciferin. We will deliver our genes to somatic cells because we want to prevent the reproduction of our GMO grass, so there will be no reason to modify the gametes. Also, in somatic cells, one tissue can be used to generate the cells of another tissue. This will be helpful if we want to reuse the plant tissue. For our quantitative analysis, we will have 2 groups. Group B will be our negative control, and Group A will be our experimental group. In our experimental group, Group A, we will have 200 GMO Kentucky bluegrass seeds that we have modified. In Group B, we will have 200 normal Kentucky bluegrass seeds. Both these groups will receive the same amount of sunlight, the same amount of water, and we will use the same type of soil to grow both. After the seedlings have matured, we will then see out of each group how many grow. And then comparing the Group A to Group B, we will see if our GMO turned out efficient. There are some technical hurdles or negative outcomes that could occur from our GMO. They could reproduce and grow into non-glowing grass areas, which then could make it difficult to distinguish pathways. It also could become invasive. Also, Agrobacterium tumefaciens are known to insert more genes than necessary into their organism.
Thank you for listening!